All right, we are wrapping up our lecture videos this week talking about blood typing. And so this is a really important concept, especially in the medical field. So one of the reasons that blood typing is super important is preventing certain types of diseases like hemolytic disease of the newborn. This is what happens when the mother's immune system ends up starting to attack the blood of the fetus during development, which is going to destroy the fetus's red blood cells and lead to all sorts of issues. This is much more common to happen in a second pregnancy, second or third, anything after that first pregnancy, because it does require that initial exposure to a certain protein, which we'll talk about in just a moment. And so it needs to kind of be exposed to that first and develop antibodies against it. And then in subsequent pregnancies, it could start attacking the baby's red blood cells. And so we'll talk about this more in a little bit, but this has to do with something called the Rh factor, which is what gives blood types their positive or negative component to those particular types of blood. And so if the mother, the person who is pregnant, has Rh negative blood, which means they do not possess this Rh protein on their red blood cells, and maybe their partner is Rh positive, so they do possess that protein on their red blood cells, and they get pregnant, and the fetus ends up being Rh positive, then the immune system of the pregnant person will produce antibodies against that protein, that Rh protein, and that's part of our immune system response, is the production of antibodies, which recognize certain specific proteins that they are trained to recognize. And so again, during a first pregnancy, that is what's happening, is we're making those antibodies, but usually the fetus is going to be unaffected completely. And so in subsequent pregnancies, though, if that baby is still Rh positive, which it most likely would be in this particular scenario, then those antibodies are already present and they are able to cross the placental barrier and get into the baby's bloodstream and start attacking those red blood cells and will destroy those red blood cells, which is obviously going to be a big problem for the baby. There is a really great, very effective treatment for this to basically prevent the pregnant person from creating these antibodies initially. And even if they miss that, there's still ways of treating it during subsequent pregnancies and stuff like that. And so there are great effective treatments for it, but that does require knowing blood types of the parents and the fetus. And so we really need to be screening for that, because the fetus would die in a case like this where the red blood cells are being attacked and destroyed. So blood transfusions are a relatively common medical treatment used for a wide variety of different purposes. They were very, very risky procedures until blood types were identified by an Austrian biologist and physician in the 1900s, early 1900s. And so before that, it really wasn't known why some people died after a blood transfusion and other people were totally fine. After we figured out that there were different types of blood based on surface proteins, it became very, very clear that there are compatible types and incompatible types. And so being able to match that is really, really important, which is why anytime somebody goes in for surgery or anything, one of the pre-op tests that's done is to type their blood so that it is known well in advance of that surgery what type of blood they are, what they need to have on hand, just in case. So there are different 4 different main blood groups that we'll get to in just a little bit, but this is an example of what a actual blood typing process might look like. And so again, the blood types are based on a surface protein. So we'll look at this slide for now. So we have ABO blood groups, and so we have 4 groups under this system: type A blood, type B, type AB, and type O, which, fun fact, actually was initially intended to be type 0, signaling that there are no surface proteins on those cells. But obviously O's and zeros can look very, very similar, and so that's how we have a type O blood, is that it was supposed to be type 0. But again, on the surface of these cells for the AB and AB groups are going to be present. So there is a surface protein type A type B surface protein, and then type AB blood has both of those proteins on the surface. This is due to a genetic phenomenon called co-dominance. So both A and B type bloods are dominant, and so if somebody has the genetics for both of those, they will express both of those surface proteins. And then type O has none of those surface proteins. In the plasma, so not on the surface of the cells, but in the plasma, there are antibodies against the other type of protein. So if somebody has type A blood, their plasma is going to have antibodies against the B proteins. So they have anti-B antibodies. All right. And the reverse is true in type B blood. In AB blood, since they have both proteins on their surface, they have no antibodies present in the plasma. Otherwise, they would attack the actual blood cells of that person. And then type O blood has no surface proteins, but both antibodies present. Okay, so the surface proteins are also referred to as antigens, and then antibodies are these other molecules that would attach to these surface molecules. So the antigens and antibodies are different things. And so this is how we determine compatibility. So coming back to this slide, so this is a blood typing test looking at using a solution containing antibodies against type A solution with antibodies against type B, and then a solution with antibodies against type D, and D represents that Rh factor, which again gives us that positive or negative identifier on our blood type. So if you have B positive blood, then you have this D factor, right, or Rh factor. So D and Rh mean the exact same thing. And so if we mix a blood sample with a solution of this antibodies against the A antigen, that A surface protein, and we get agglutination, which is clumping of the red blood cells shown here in this image, so that's what's happening here, that tells us it is that type of blood. I know that's kind of confusing to think about it that way, but again, coming back to this picture, A antigens on the surface, but antibodies against B. So if we were to put type B blood into this person with A blood, then these antibodies would attach to these surface proteins on the B blood cells and make them all clump together, which is going to form blood clots inside of the blood vessels, in the tissues, and cause all sorts of issues, usually leading to death. So this is really, really dangerous, and why blood types need to be identified. All right, so this is what agglutination looks like. It's all of this clumping, and so if we don't see that, it means we are not looking at that type of blood. So for this particular test right here that we're seeing on the screen, this blood type is A positive. So this clumping means it is that type of blood. So we have clumping in the antibodies against A, which tells us the anti-A antibodies bound to A surface proteins, causing that clumping. We don't have type B proteins there, so nothing happened in this case. And then we have clumping in the anti-D antibodies or the anti-RH factor, and that tells us that protein is present, giving us a positive. Okay, so that is how we type blood. It's a pretty quick process, and there's a few different ways of doing it, but it's a really, really quick, easy process. But in like a hospital setting or something, because we really don't have room for error with blood typing, we do not want to transfuse the wrong type of blood. Usually what will be done in a medical setting like a hospital is one lab tech will come through and type the blood sample and get their results and mark that down, and then a second lab tech will come through and type that blood and record their results. Usually this is typed a minimum of 3 times to eliminate any potential issues. And as long as all 3 people get the same results, you can pretty safely assume that those are correct results and you have the right blood type identified. If each person got different results, then there's clearly an issue going on. Somebody did something wrong. Something's wrong with the blood sample. Various things like that. And they need to do a lot more checking to see what's going on. All right, so agglutination in the actual blood vessels is very, very dangerous, will cause organ failure, and usually lead to death, especially if we're transfusing like a whole bag of blood. That's going to be a lot of blood cells coming in and a lot of those antibodies coming in. All right, and so that's really, really bad. After those clumps break down, this is called hemolysis or hemolysis. That is, the cells are going to actually start breaking down. This can cause issues in the kidneys because of the amount of hemoglobin being released, because so many cells are being broken down simultaneously, and that can lead to kidney failure. So this is really, really dangerous to have the wrong type of blood transfused. So going back to our groupings here, this last row down here, you can see the blood type compatibility. So if somebody has type A blood, they can safely receive blood from another type A person or a type O person, because again, that type O person is not going to have any of those surface molecules. Frequently, the plasma, especially of type O blood, can be filtered to remove some of these antibodies. But even if it's not, there's not as much of a risk for the antibodies in that sample to react, because there's going to be fewer of them present. But it is also common to have some of those filtered out in certain scenarios. Type B blood can receive, again, type B blood, obviously, and type O. AB blood can get blood from A, B, and O. So that is pretty universal as a recipient. And this is without counting in the RH factor, the positive and negative. And then for type O blood, they can only get blood from type O. And so they are considered to be the universal donor. So type O blood can give blood to anybody, but it can only receive blood from other type O. All right, so we have universal donors and universal receivers. So if we get in that, whoops, sorry, too far. If we factor in that RH, which again will give us the positive or negative, it's going to function similarly, where there's either going to be RH proteins on the surface of the blood cells and then no antibodies, or there are going to be no RH proteins and there will be antibodies against the RH proteins. And so that's again going to give that positive and negative component, and that's another factor to consider. So coming back to that hemolytic disease of the newborn, in the case of exposing somebody who is Rh negative to an Rh positive scenario, they will make those antibodies against the Rh protein and start attacking those Rh positive cells. All right, so that positive and negative needs to be factored in as well. Okay, so the negative, anything negative, is going to be able to safely donate to a positive, but we cannot donate from an Rh positive person to an Rh negative because that will cause a reaction. And so that is why AB positive blood is the universal recipient. So it can get A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, or O negative blood. It can get any blood type regardless of ABO group or Rh factor. O negative is the universal donor, so it will be able to give blood to literally any other type as long as it is OH or O negative. All right, because it will not have that Rh factor, so it would not have any of those surface proteins on it. Everything else gets a little bit more specific. So that's why people who are O negative blood type, if they've donated blood once and it is on record that they are O negative, frequently they will get calls at the 3-month mark, which is how long you have to wait in between blood donations, saying, hey, it's been 3 months. We could really use another donation because, in an emergency scenario, for example, when you need to transfuse blood, you need to give that patient blood. You do not have time to type their blood and figure out what their blood type is. They will be getting O negative blood. All right. There are other surface proteins on the cells that can play a role, just not to as great of an extent, so we don't really focus on those as much in the medical setting, at least at this point in time. We may discover at some point down the road that some of those are way more important than we thought, but as far as we can tell right now, that ABO group and then the Rh factor are the most important components to consider when doing blood transfusions. This also goes for organ donations and those bone marrow transplants. All right, and so that is all about blood typing. I think it's super interesting, especially when you start getting into those genetics and stuff like that. Like I said earlier, A and B are both dominant genetics, whereas type O is the recessive, which is how we get that AB type blood, is both of these genes will get expressed simultaneously on the same cells. So I've always thought that's really, really interesting. But this is why blood typing is so absolutely essential. And being able to always have that O negative blood on hand as well, especially in case of an emergency. And that wraps up our lecture videos for this week.